Hi, I'm Dr. Cecily Bishop, and I am the chair of the SSR Public Affairs Committee, and I am joined today by Dr. Jon Hennebold, who's the professor in chief of the Division of Reproductive and Developmental Sciences at the Oregon National Primate Research Center. And I've asked Jon here today to talk to us about gene editing in primates.
Hi, Cecily. Thank you for having me. It's a pleasure to talk to you today.
So when we talk about gene editing in primates, I really wanted to talk about what do we look at for the application of these techniques when we're talking about specifically to primates?
So what we're focusing on is using new gene editing technologies. And when I speak of gene editing techniques or methods, particularly the newer varieties that have developed over the last few years, I'm talking about CRISPR-Cas9 or TALENs. Primarily CRISPR-Cas9, or people typically refer to it as just CRISPR. So the ability to modify a primate genome is now possible through this new technique. And I think that really, to understand what a huge advance this is, we have to go back and look back what we've been able to do over the last several decades. And if I would have mentioned this technology or the possibility of editing a mammalian genome, most people would have thought of homologous recombination or creating knockout mice. 5 years ago, 10 years ago, this was the standard for trying to understand how genes function. And in this process, you are able to create a mutation within a targeted gene in embryonic stem cells, transfer those stem cells to mouse embryos, and then transfer the embryo to a surrogate female, which will then provide pups that will carry this mutation. Through subsequent breeding, you can generate genetically identical animals, with the only difference being that they have a mutation in a gene of interest. And this has allowed us to understand how genes function. It was a huge advance for molecular and cellular biology. And in fact, it resulted in 3 Nobel Prizes awarded to the individuals who basically developed this technology. Now, the thing is that we could never apply this to primates because, one, the generation time, the time to sexual maturity, and differences in embryonic development have prevented this technology from being applied to species outside of rodents, primarily mice. So this was a game changer, basically. It now allows us to target embryos directly so that we can look at genes that cause specific human diseases, so that now we have disease models to understand how the disease develops. But more importantly, we can also try to now create ways to treat those diseases, to prevent them from progressing, or maybe even to correct them at the genetic level.
So when we talk about these, and we really want to talk to you about the success rate. So I know rodents are the gold standard. They have the highest success rate. So what are we talking about as far as efficiency and efficacy in primates?
Yes, so you're correct, because rodents have always been much easier to manipulate in terms of mutations within the genome. So as I mentioned, in homologous recombination, within a few generations, you can have, again, pretty much a genetically homogeneous population of animals. In terms of CRISPR or TALENs, the ability to create mutant lines is commonplace now. But before we talk about that specifically, we really should take a step back and talk about the types of editing that can occur with the new technologies. So in terms of CRISPR-Cas9, the embryo or the zygote or the cells are provided nuclease, which is the Cas9, and then a guide RNA that directs Cas9 to a particular target gene. Once it binds to that site in the genome, it creates a double-stranded DNA break, and the cellular machinery does not like to have a double-stranded DNA damage or break being present, and so it will rapidly try to correct that double-stranded DNA break. And what it does it at a low level of fidelity. So it either adds or deletes bases. So the downstream effect is that it creates a frame shift in the protein that's produced, which then leads to a non-functional protein and a specific type of mutation. Now, these can be rather random in terms of the number of nucleotides that are added or deleted, and it can also be different in terms of what happens on one allele versus the other allele. So it's a rather random event. Now, to overcome the randomness of Cas9 along with the guide RNA by itself, you can provide a DNA template. And if there's a DNA template present, then it will use that template to repair the double-stranded DNA break. And it will incorporate whatever you have included within that DNA template to be integrated into the genome. So if you want a particular mutation or if you want a very specific change in the DNA, then you incorporate that into your DNA template. So if talking about, in terms of talking about the random approach without a DNA template, that's called non-homologous end joining. And then if you add a DNA template to add a specific mutation, that's called homologous directed repair. In terms of non-homologous end joining in primates, the efficiency has increased and just continued to increase over the past several years, to the point where now we can introduce mutations in non-human primate embryonic genome levels of probably 50 to 75%. In terms of HDR, that's where the big difference lies between primates and rodents. Rodents are much more effective in that regard, and in fact, numerous mutations have been created, specific mutations have been created in mice using HDR, but yet in primates, that efficiency is only between 5 to 20%. So there's something very different in terms of the underlying cellular mechanisms that allow for the integration of a specific mutation via the HDR approach.
Okay, so when we talk about this, we do see a lower efficiency. Really, when we look, though, at the field seems to try to be driving forward, especially with human gene editing. And specifically, I'm talking about the two cases, the one scientist in China who had announced that he had the live birth of a baby girl with the mutation for the gene, one of the genes important in viral entry for the HIV/AIDS virus. And another scientist in Russia who said he's performed similar experiments but is waiting for an approval before he implants those embryos. So what do you think are really the big ethical questions at this point when we talk about taking these into the human genome?
Well, I think first and foremost, the biggest issue relates to safety. And at present, the concern is that the gene editing reagents that are used have the potential to bind to other regions within the genome, the region outside of the gene that you're trying to mutate or you're trying to fix, may be susceptible to gene editing, and thereby leading to other very significant effects in terms of cellular function. So it's really this off-targeting level that we don't have a good understanding of, and has the potential to lead to more significant issues than the underlying disease. And I think that's particularly relevant with the genes that have been targeted in humans. They relate to HIV infectivity, and so they're not associated with early death or significant morbidity or mortality through the natural course of development. Very important for HIV infectivity, but even there, it's not 100% dependent on the presence of this gene for the virus getting into the cell. So the underlying target was not well justified. Then you have on top of that potential off-target editing effects that could have serious deleterious effects. And collectively, it just points to the fact that this technology is not ready for clinical use, and that is the general consensus of the scientific community. There is one other factor too that was not really taken into consideration, and that is the possibility of mosaicism, where you have editing that occurs after cell division, after fertilization. And so you may end up with different cell lineages with different levels of editing and different levels of off-target effects. And so you may have differential sequestration of those edits within different tissues, and we don't know what that means in terms of health and safety issues.
Yeah, so when we talk too about those off-target effects, I wanted to get your thoughts on this new technology that's recently come out, the prime editing, where they say that now they can actually make some more precision editing of the genome. So what are your thoughts on this newer technology?
Well, it was a remarkable advance in the field by increasing the level of specificity, getting to the problem that we were just referring to. And it really has the potential to limit these off-target effects. However, as with everything else, and again, getting to the ethical concerns, it's been demonstrated in cell lines and in other models, but we don't really understand what the broad range off-target effects of this technology is, even though it appears greater or more selective than the other, the previous iterations. It's a step in the right direction. So I think the important thing to take from this is that the field is evolving so rapidly. If we were here 5 years ago, the whole field would just be in its infancy in terms of CRISPR, TALEN, mediated gene editing, and the possibility was just unfolding, and that's a relatively short period of time. And here we are with this new editing system and increased specificity. So that was a very short turnaround time in terms of developing the system to advancing to refining it. And within a year, 2 years from now, who knows what further refinements will be in place. We are headed in the right direction. We're clearly trying to limit these off-target effects so that who knows, we may be closer to having something that's more appropriate for clinical use.
So right before we wrap up, I'd just like to ask, before we end, about your thoughts about the value of the non-human primate model for these type of technologies and these type of experiments, really the preclinical value that you see that this model can offer.
Well, I think that we've already hit the important points a couple of times, and I think that, for example, homologous recombination and utilizing that technology, how it was restricted to rodents because there's such a difference in terms of the underlying biology that allows for DNA repair processes, for propagating mutations, for correcting mutations. The efficiencies are considerably significantly different between the species. Couple that to the difference in the genomic sequence, the divergence, the physiological divergence, the how the reproductive processes differ. It doesn't replicate what happens in humans very well in a lot of regards. Even though it was a rodents and knockout mice have provided us tremendous amount of data in terms of how genes function. In terms of using technologies, we have to understand safety and efficacy, and that's why we need to utilize the non-human primate model. It will give us insight into how effective it is, and then also what our potential off-target effects are and what that might mean in terms of problems from developing a particular technology.
So thank you so much for being here today, Jon Hennebold, and talking to us about this. And we look forward to talking to you again in the future.
Thank you. It's been a pleasure speaking with you today.